And it's my pleasure to introduce Santosh. He is a second-year PhD student in the Comparative Medicine and Integrative Biology program at Michigan State University's College of Veterinary Medicine. And he's working in the MSU's fish pathobiology and immunology laboratory under the guidance of Dr. Bart Gorglione. So, take it away, Santosh.
Yeah, thank you. So for today's talk, I'm going to talk about the cyprianid herpes virus 3, also known as the koi herpes virus, and the carp virus in the invasive Asian carp species from the Illinois River. So briefly describe about the what do we mean by Asian carps when we talk about that. So it includes 4 species: grass carp, silver carp, black carp, and the big egg carp. So they introduced into the US region in the 60s and 70s, but later they escape into the natural waters either because of the flood or because of the intentional or unintentional human activities. So why they're the species of concern? Because they have the very high fecundity and they can adapt the wide range of environment. So because of that reason, they can compete with the native fish from this region for the food or for the shelter or also for the breeding grounds. So currently, we don't know whether they can carry some of the pathogens and they can transfer to the other fish or not. So that is also one of the potential concern. And together, all these reasons, they are threatening the Great Lakes. Once they enter the Great Lakes, they can do a lot of damage, and it is the irreversible process. You cannot control them because of those characteristics of those fish. So currently, there has been a lot of actions going on between collaborating different states of the US and the Michigans. Those includes the deterrent technology using the certain electric or sound barriers, and also the seasonal mass removal campaigns in the Illinois River. So that is where we are most interested in, because they are using the large net to remove the lot of samples, and those samples are discarded. So we got opportunity to go get these samples and then test for the pathogens. And also some other organizations are doing eDNA sampling or other kind of surveys to detect those fish. So what is the briefly about the koi herpes virus? So it is the cyprianid herpes virus 3, one of the largest herpes virus with 295 kilobase per genome. So the main clinical signs of this virus includes necropsis in the gills and also the ulceration of the skins. So within the water range of 18 to 25 degrees Celsius, the disease can happen, and then the mortality can reach up to 80 to 90%. So as with any other herpes virus, the latency is also the characteristics of this herpes virus. Whenever the temperature is not suitable or other kind of environmental condition, they can go into the latent stage, and again within the optimum range of the environmental conditions, they can the disease can again happen. So they first noticed in the USA and Israel and UK in the 90s, and then but after that, this virus is already been circulating in the Midwestern states. And if we talk about the Michigan, so first detected in the koi pond near the Grand Rapids, and then there has been several outbreaks of the because of this virus, causing a lot of common carp killing and the dying, so causing the huge attention among the officials and the media as well. So it is likely that this virus has been spreading and then causing a lot of issues for the naturalized common carp population that has been already established in this region. And it can cause a lot of troubles for the communities that depend on the lakes and the ecosystem, as well as the anglers who are who want to do the recreational fisheries. So talking about the carp virus, which is another virus, one of the largest pox virus that infect the vertebrates. So around 450 kilobase pair of the DNA. The genome. And then this virus is first detected in the Japan in the koi carp. So after the first detection, 20 years after the first detections, they also found in the US, because US import a lot of ornamental koi carp from the outside. And the clinical signs are especially the gill damage is kind of similar to the external signs are similar to the KSB. And the typical characteristic is that the sleeping or sleeping behavior, like coma-like behavior when the fish get infected. And also the oedematous conditions, the swelling in the internal organs is also this typical characteristics of this virus. And it also causes the huge mortalities in the koi found at the feral carp. So this virus is also rapidly circulating around the world, and then also depends on the stress conditions and the seasonality. So most of the cases, this virus found with co-infection with the KSV or the CYSV3, and this virus can be a new notifiable disease very soon. So talking about the Illinois River sampling. So basically, we did the Illinois River sampling in the summer of 2021 and 2023. So we took the tissue samples for molecular biology confirmation and virology and histology. So these are some pictures. So during the harvest, we got they discard the phase. So we got the opportunity and we get the tissue sample from those phase. So our aim for this research or to get these samples is to study the occurrence of those two viruses among the invasive phase and also the native phase of that region. So by doing that, we want to isolate the local strains of KSV so that we can do the virological analysis. So, and then we can define using those local strains whether the invasive Asian carp region here are susceptible to that virus or not. So here is a descriptive samples that we got. So from summer 2021, we got 38 phase, including silver carp, bigot carp, common carp, smallmouth buffalo, and freshwater drum. And in 2023, we get much more number than that, like 152 total. So we also got some big mouth buffalo and grass carp as well. And for the molecular examinations, we use the pooled tissues of 20 milligram, including gills, kidney, liver, and spleen. And we use that for DNA extractions. And we use the conventional PCR or the quantitative PCR for the detection of the both KSV and the CEV. And also for the sequencing and the confirmation of the presence of those viruses. So results from 2021 samples, we can see like 11 total phase we got KSV positive, and then 12 got CEV positive. So if you look at the distribution of the viral load among the species, they are varies slightly. So CEV load is a little bit higher than the KSV, but again, it varied based on the species. And so the most importantly is we detect the KSV in the smallmouth buffalo. So smallmouth buffalo is one of the cypriniformes, but and this is really interesting for us because this virus hasn't been detected in this species before. And also from the Illinois River, we also get the KSV in the silver carp as well. So these together from those samples, we detected CEV for the first time also in the Great Lakes regions. And interestingly, 3 small buffalo, they were also co-infected with the CEV, both CEV and KSV. So that's also important, considering those are the important native phase in this region. And from the 2023 samples, so we got a lot of phase, and we our detection of the KSV and the CEV both happens in species like silver, pighead, and freshwater drum, and smallmouth and big mouth buffalo as well. So previous samples we got from the silver carp, and these samples we got also from the bigot carp as well. And also the freshwater drum, so which is not, so it's not a species from the order Cypriniformes, but it is also one of the important ecologically or economically important in this region, the native phase. So we also get detection from that as well. And also our results also shows like consistent with our previous detections, because we again detect from both the native and invasive species. So, and one of the freshwater drum were co-infected. So taking together like native phase, they are being detected with the presence of both the virus, and some of them are co-infected, and invasive carp as well. So we did some sequencing of the especially DNA polymerase for the KSV and the viral protein P4A for the CEV, and some phylogenetic analysis. So we found that the KSV sequence that we got from our samples are closer to the European genotype, whereas for the CEV, it is belongs to the genogroup second. So we try to isolate the virus from our samples. So for that, we inoculated them with the homogenate of the gills, spleen, and kidney. We didn't include the liver in this virological analysis. So, and we included them for the conventional cell lines like KF1, CCB, and also our newly developed grass-carp fin cell lines. And putting together, incubate them for 14 days at 25 degrees Celsius, but we didn't observe the visible cytopathic effect. So, and so we kind of unable to isolate the virus. And when we do the mRNA specific PCR as well, so we're not also able to detect the presence of the mRNA. So likely because there might be the latent stage of the CYSV3, because when they're in the latent stage, it's difficult to isolate the virus. And also CEV, in the case of CEV, they are still not culturable in the cell line. So we didn't try for that CEV. So in summary, so this is the first detection of both ASV and the CEV in the Asian carp and the native species from the Illinois River. So, and the first report of co-infection of both viruses in the native species like smallmouth buffalo and freshwater trout. So these native phase, they haven't like moths publications or the research related whether they are in their pathology or in response of their response in terms of pathogens. So this is quite interesting considering their importance in this region. So especially the big mouth buffalo, they live like really long. So one latest publication show that they can live up to 127 years. So considering those facts, so whether those fish can be infected with these two viruses, or if not, then they can carry some certain time and transfer with the other fish or not, or how these fishes, both Asian carp and native species, can actually change the disease dynamics and disease ecology of this region is really interested. And then it's kind of opened the door for like they might infect it or they might carry the virus. So further studies are needed for that. And it's important to include the CEB as well. So yeah, and I'd like to thank the BART project and the Illinois department and my lab member as well, and Michigan State University. Thank you.
Great presentation, Santosh. That's important work, identifying the host range of those two viruses. Folks, if you have a question, we have just a minute here.
Excuse me?
Yeah, go for it, Jesse.
Hi, thank you. Yeah, I have one question. So I noticed like you try to re-isolate the KHV, the CYHV3 from the fish, and then to try to re-isolate it by using different cell lines. I noticed like you use like a gill or some other spleen and kidney, right? Did you try other tissues like a brain?
So yeah, for the brain, like we didn't get the brain tissues from our last sampling. So if they're in the latent stage, they might be in the brain. I mean, it's just proved to be in the brain cells. So we're definitely planning to get some of the brain tissues from our next sampling, so that we might get the virus as well.
Yeah, because the herpes virus, they can affect many type of cells, but the major one, so they can affect the nerve system, like the target tissue, like a brain more than the kidney or spleen. I think you may want to try brain, and also like you will get low toxic.
We got the cytotoxic effect, so that's why one reason we don't want to include the liver as well. But yeah, brain is definitely one of the interesting tissues that we're looking for. Thank you.
Yeah, thank you.
Santosh, one last quick question here. Apologies if I missed it. Can you talk about the mRNA testing that you did and whether or not the virus was found to be actually replicating, or if it was just
yeah, so we try to see whether the virus are replicating or not. So that's why we tested the mRNA specific sequence. So we use a ORF89 sequence for also for the mRNA detection.
Okay, and were those positive or negative?
they were negative, which kind of says that the virus are not replicating. So
Were they negative in the common carp host too?
so they were negative in the common carp as well in the both of the samples. So which kind of shows that they might be in the latent states, or they're not active the time that we got those samples. So that is why we are continuing to get more samples and diverse our analysis. So we'll know very soon that how is the status.
That's great. Esteban, I see you got your hand up. We're over time, so if you've got a quick one, you can go for it.
Sure, it's two quick ones. So the first one, Santosh, so great job, by the way, but did you mention the temperature at the time of collection? And then the second one was, I noticed that for some of your qPCRs, the standard error or standard deviation, I don't know if you were presenting standard error or deviation, was pretty big. What is your cutoff? What cutoffs are you using for the CT values, for your qPCRs? Because for some of the drums and some of them, they're pretty big. So I'm not sure if, the detection limit is an issue.
Yeah, so for the temperature, so we collected those samples in the summer. So water temperature probably should be around 20, 22, 25. So we haven't measured water temperature last sampling, but this year we got all the equipments and everything. So we're going to do that as well. So that we'll know that the temperature whether plays some role in that or not. So, and for the qPCR, so we generated the standard curve, and by the standard curve, the cutoff value is 38. So after the 38, we consider them as a negative after the 38. So that is the standard curve. So the change in the graph, as you mentioned, that the big change is because for that big change, I think those samples are only 3 samples positive. So that's why the deviation is high.
All right. Thanks a lot, Santosh. Folks can follow up with you directly if they've got more questions. Appreciate the overview of your work.